Welcome dear students. Today we are going to discuss the secondary messenger concept. The brief objectives of today's lecture are to get an overview of hormones, to understand the hormone receptors and their activation. We shall also try to highlight the regulation of the number and sensitivity of hormone receptors, followed by explaining the secondary messengers concept in detail. And finally, we shall talk about the different types of secondary messenger systems. Let us first talk about hormones. Hormones are small molecules or proteins that are produced in one tissue, released into the circulation and carried to other tissues, where they act through receptors to bring about changes in cellular activities. They serve to coordinate the metabolic activities of several tissues or organs. Virtually every process in a complex organism is regulated by one or more hormones. For example, maintenance of blood pressure, blood volume, and electrolyte balance, embryogenesis, sexual differentiation, development and reproduction, hunger, eating behavior, digestion and fuel allocation. We examine here the methods for detecting and measuring hormones and their interaction with receptors, and we consider a representative selection of hormone types. The coordination of metabolism in mammals is achieved by the neuroendocrine system. Individual cells in one tissue sense a change in the organism's circumstances and respond by secreting a chemical messenger that passes to another cell in the same or different tissue, where the messenger binds to a receptor molecule and triggers a change in this second cell. These chemical messengers may relay information over very short or very long distances. In neuronal signaling, as you can see in the figure, the chemical messenger is a neurotransmitter, for example, acetylcholine, and may travel only a fraction of micrometer across the synaptic cleft to the next neuron in a network. In hormonal signaling, the messenger hormones are carried in the bloodstream to neighboring cells or to distant organs and tissues. They may travel a meter or more before encountering their target cell, as you can see in the figure. Except for this anatomic difference, these two chemical signaling mechanisms are remarkably similar, and the same molecule can sometimes act as both neurotransmitter and hormone. Epinephrine and norepinephrine, for example, serve as neurotransmitters at certain synapses of the brain and neuromuscular junctions of smooth muscle, and as hormones that regulate fuel metabolism in liver and muscle. Let us have a look at hormone receptors and how they are activated. The first step of a hormone's action is to bind to specific receptors at the target cell. Cells that lack receptors for the hormones do not respond. Receptors for some hormones are located on the target cell membrane, whereas other hormone receptors are located in the cytoplasm or the nucleus. When the hormone combines with its receptor, this usually initiates a cascade of reactions in the cell, with each stage becoming more powerfully activated so that even small concentrations of the hormone can have a large effect. Hormonal receptors are large proteins, and each cell that is to be stimulated usually has some 2000 to 1 lakh receptors. Also, each receptor is usually highly specific for a single hormone. This determines the type of hormone that will act on a particular tissue. The target tissues that are affected by a hormone are those that contain its specific receptors. The locations for the different types of hormone receptors are generally the following. First one is the in or on the surface of the cell membrane. The membrane receptors are specific mostly for the protein, peptide and catecholamine hormones. The second type of receptors are that are in the cell cytoplasm. The primary receptors for the different steroid hormones are found mainly in the cytoplasm. Third one is the receptors that are present in the nucleus of the cell. The receptors for the thyroid hormones are found in the nucleus and are believed to be located in direct association with one or more of the chromosomes. Now, let us have a look at the regulation of the number and sensitivity of hormone receptors. The number of receptors in a target cell usually does not remain constant from day to day or even from minute to minute. The receptor proteins themselves are often inactivated or destroyed during the course of their function, and at other times they are reactivated or new ones are manufactured by the protein manufacturing mechanism of the cell. For instance, increased hormone concentration and increased binding with its target cell receptors sometimes causes the number of active receptors to decrease. This down regulation of the receptors can occur as a result of, first one is the inactivation of some of the receptor molecules. The second reason can be inactivation of some of the intracellular protein signaling molecules. And the third one, temporary sequestration of the receptor to the inside of the cell, away from the site of action of hormones that interact with cell membrane receptors. The fourth one is destruction of the receptors by lysosomes after they are internalized. Or fifth one, decreased production of the receptors. In each case, receptor down regulation decreases the target tissue's responsiveness to the hormone. Some hormones cause upregulation of receptors and intracellular signaling proteins, that is, the stimulating hormone induces greater than normal formation of receptor or intracellular signaling molecules by the protein manufacturing machinery of the target cell, or greater availability of the receptor for interaction with the hormone. When this occurs, the target tissue becomes progressively more sensitive to the stimulating effects of the hormone. Now, let us have a look at secondary messenger mechanisms for mediating intracellular hormonal functions. One of the means by which hormones exert intracellular actions is to stimulate formation of the second messenger, cyclic AMP, inside the cell membrane. The CAMP molecule then causes subsequent intracellular effects of the hormone. Thus, the only direct effect that the hormone has on the cell is to activate a single type of membrane receptor. The second messenger does the rest. Cyclic AMP is not the only second messenger used by the different hormones. Two other especially important ones are calcium ions and associated calmodulin, and second one is the products of membrane phospholipid breakdown. Now let us have a look at the adenylyl cyclase cyclic AMP second messenger system. The table displayed here shows a few of the many hormones that use the adenylyl cyclase CAMP mechanism to stimulate their target tissues. And the figure displayed here shows the adenylyl cyclase CAMP second messenger system itself. Binding of the hormones with the receptor allows coupling of the receptor to a G protein. If the G protein stimulates the adenylyl cyclase CAMP system, it is called a GS protein, denoting a stimulatory G protein. Stimulation of adenylyl cyclase, a membrane-bound enzyme, by the GS protein then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate or ATP into CAMP inside the cell. This then activates cyclic AMP dependent protein kinase, which phosphorylates specific proteins in the cell, triggering biochemical reactions that ultimately lead to the cell's response to the hormone. Once cyclic AMP is formed inside the cell, it usually activates a cascade of enzymes. That is, first one enzyme is activated, which then activates a second enzyme, which then activates a third, and so forth. The importance of this mechanism is that only a few molecules of activated adenylyl cyclase immediately inside the cell membrane can cause many more molecules of the next enzyme to be activated, which can cause still more molecules of the third enzyme to be activated, and so forth. In this way, even the slightest amount of hormone acting on the cell surface can initiate a powerful cascading activating force for the entire cell. If binding of the hormone to its receptors is coupled to an inhibitory G protein denoted by GI protein, adenylyl cyclase will be inhibited, reducing the formation of cyclic AMP and ultimately leading to an inhibitory action in the cell. Thus, depending on the coupling of the hormone receptor to an inhibitory or a stimulatory G protein, a hormone can either increase or decrease the concentration of cyclic AMP and phosphorylation of key proteins inside the cell. The specific action that occurs in response to increase or decrease of cyclic AMP in each type of target cell depends on the nature of the intracellular machinery. Some cells have one set of enzymes and the other cells have other type of enzymes inside their cytoplasm or nucleus. Therefore, different functions are elicited in different target cells, such as initiating synthesis of specific intracellular chemicals causing muscle contraction or relaxation, initiating secretion by the cells and altering cell permeability. Thus, a thyroid cell stimulated by cyclic AMP forms the metabolic hormones thyroxine and triiodothyronine, whereas the same cyclic AMP in an adenocortical cell causes secretion of the adrenocortical steroid hormones. In epithelial cells of the renal tubules, cyclic AMP, for example, increases their permeability to water. Now, let us have a look at the cell membrane phospholipid second messenger system. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C that is attached to the inside projections of the receptors. This enzyme catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidyl inositol biphosphate or PIP2 molecules, into two different secondary messenger products, inositol triphosphate or IP3 and diacylglycerol. The IP3 mobilizes calcium ions from mitochondria and the endoplasmic reticulum, and the calcium ions then have their own second messenger effects such as smooth muscle contraction and changes in cell secretion. Diacylglycerol or DAG, the other lipid secondary messenger, activates the enzyme protein kinase C or PKC, which then phosphorylates a large number of proteins, leading to the cell's response, as you can see in the displayed figure. In addition to these effects, the lipid portion of diacylglycerol or DAG is arachidonic acid, which is the precursor for the prostaglandins and other local hormones that cause multiple effects in tissues throughout the body. Now, let us have a look at calcium calmodulin secondary messenger system. Another secondary messenger system operates in response to the entry of calcium into the cells. Calcium entry may be initiated by first changes in membrane potential that open calcium channels. Second, a hormone interacting with membrane receptors that open calcium channels. On entering a cell, calcium ions bind with the protein calmodulin. This protein has 4 calcium sites, and when 3 or 4 of these sites have bound with calcium, the calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinases. Activation of calmodulin dependent protein kinases causes by phosphorylation, activation or inhibition of proteins involved in the cell's response to the hormone. For example, one specific function of calmodulin is to activate myosin kinase, which acts directly on the myosin of smooth muscle to cause smooth muscle contraction. The normal calcium ion concentration in most cells of the body is 10 raised to the power minus 8 mole per liter, which is not enough to activate the calmodulin system. But when the calcium ion concentration rises to 10 raised to the power minus 6 mole per liter, enough binding occurs to cause all the intracellular actions of calmodulin. This is almost exactly the same amount of calcium ion change that is required in skeletal muscle to activate troponin C, which causes skeletal muscle contraction. It is interesting that troponin C is similar to calmodulin in both function and protein structure. Dear students, it was all about today's lecture. Hope you have understood it well. Thank you and goodbye.